Okay, finishing up today and probably next Tuesday with a little bit more on modern synthesis evolution. I know we talked about inbreeding on, wow, what day was it? Tuesday. So some of this is just going to be review. We talked about how we can define alleles in two different individuals as identical by descent if they descend from a recent common ancestor. My alleles for brown eyes and my sister's alleles for brown eyes would have to be identical descent because we both got them from my dad. On the other hand, my alleles for brown eyes and the lead singer of BTS's alleles for brown eyes are not identical by descent because we don't have any recent common ancestors. I'm reasonably sure about that. Now, the fraction of alleles in a population that are identical by descent is called capital F. That's the inbreeding coefficient. And we talked about that, and I showed you a simple way to calculate that. F is equal to 1 minus the observed heterozygosity over the expected heterozygosity. I want to derive that in just a little bit more detail. Imagine that we're back in our imaginary world of oysters that are all firing off eggs and sperm that have each one of which carries either a big A or a little a allele. The oyster world we've talked about before. So if we have this pool of eggs and sperm, the probability that we're going to pull out an egg, let's say, with the big A allele is some number between 0 and 1, and we can call it P. That's the frequency of the big A allele. And Q is going to be the frequency of the little a allele. The probability that you reach in and pull out an egg with the big A allele is P. The probability that you reach in and you pull out a sperm that has the allele that is identical by descent is F. So the probability of that any given zygote is going to have the genotype big A, and its alleles are identical by descent is PF. The probability of getting a big A zygote with alleles that are not identical by descent is P, the probability that the egg will have a big A allele, times P, which is the probability that the sperm will have a big A allele, times 1 minus F, which is the probability that the alleles aren't identical by descent. I'm not going to make you duplicate these calculations on the exam, but I do want you to at least get some sense of where I got that formula that I brought up on Tuesday. You can show in the same way that the probability of getting a little a zygote with alleles that are not identical by descent is Q squared times 1 minus F. And the probability of getting a heterozygote turns out to be 2PQ times 1 minus F. Now, if there's no inbreeding going on, then F is equal to 0, and 1 minus F is 1, and we're back in Hardy-Weinbergville. These equations become identical to the predictions we'd make under Hardy-Weinberg. Aaron, Aaron. Okay, sorry, unscheduled appearance from my kid. Okay, so in a population where we're looking at inbreeding, the total frequency of big A genotypes is the sum of those two formulas, PF plus P squared times 1 minus F. And we can do a little bit of algebra on this. We can rearrange it as you see, and then since Q is always equal to 1 minus P, we can turn that into p squared plus pqf. So if there is significant inbreeding going on, then the frequency of big A genotypes is p squared plus pqf. The frequency of little a genotypes is q squared plus pqf. And the frequency of heterozygotes is 2pq times 1 minus f, which you can expand as 2pq minus 2pqf. So remember that the signature of an inbred population is increasing number of homozygotes, and decreased numbers of heterozygotes. Specifically, the fraction of each homozygote is going to be higher than would be predicted under Hardy-Weinberg equilibrium, and PQF gives you that proportion of excess homozygotes. Okay, the observed frequency of heterozygotes is HO. Thank you, love. And that's going to be 2pq times 1 minus f. The expected frequency of heterozygotes, if there wasn't any inbreeding, is just 2pq. And that's how we get that formula that f is equal to 1 minus HO over HE. 1 minus the observed homozygote frequency divided by the expected heterozygote frequency. I'm not going to expect you to derive that, but this is ultimately where it comes from. And you do need to know that formula for calculating the inbreeding coefficient. Okay. Here's the thing. We use this in artificial selection a lot, because if you're creating a breed of animal or strain of plants or something like that, what you want is for all individuals to be identical in important ways, right? If you've got a very fast racehorse, what you want is to have that, that racehorse make lots of babies that will also be very fast racehorses, right? You don't want to breed your racehorse and get Clydesdales, very nice horses, I'm told, but that's not what you want. By the same token, if you've got a very good guard dog, you want that guard dog to have puppies that all grow up to be guard dogs, because that's valuable. That's what you're after. And so when breeders are creating a breed or a strain of animal or plant, it works in plants as well, they'll use inbreeding to fix desired traits, because what you want is a bunch of horses or a bunch of Dobermans or a bunch of whatever species it is that are all homozygous for the genes that you want to have. You want a bunch of racehorses that are all homozygous for all of the genes that contribute to speed. Everything from genes that affect leg length to genes that affect oxygen physiology, right? You don't want to breed two expensive racehorses together and get a Shetland pony. That's not what you're in the business for. And so if you go back and look at the pedigrees of many of our domesticated animals, they consist of an awful lot of sibling matings or, father-daughter matings and things like that. Thoroughbred racehorses, by definition, the official definition is they have to be descended from one of 3 Arabian racehorses that were sent to England in the 1960s. They have to be descended from the Godolphin Arabian, the Byerly Turk, and a third one whose name I cannot recall at this moment. And each one of them only mated with a limited number of mares. If you are Yeah, if you are racing thoroughbred horses, thoroughbred means that they are descended only from those three males and a limited number of females that have been brought in. And that means that, thoroughbreds are practically genetically identical. There's no way to get around the fact that they have all been very heavily inbred. This is why in domestic dogs, inbreeding causes an increase in desirable trait homozygosity, but undesired traits can also increase in frequency because of increased homozygosity. This is why just about every domestic dog breed has some kind of genetic problem that tends to be common in it. Doberman pinschers are susceptible to getting a bleeding disorder called von Willebrand's disease. Dalmatians are prone to deafness. The solution for that is you inbreed, and then you pick out the individuals that are homozygous for what you want and destroy the individuals that are homozygous for what you don't want. If you're a Dalmatian breeder, you're expected to humanely put down any of your Dalmatians that are deaf. You're not even supposed to have them spayed or neutered and give them away as pets. You're supposed to make absolutely sure that they don't breed. So the upshot is that you have Dalmatians that are homozygous for all of the genes that give them a high energy disposition and attractive spots. But then you use selection to get rid of the Dalmatians that are homozygous for traits like deafness. Not necessarily a particularly nice way to do business, but this is what you do if you're developing breeds. Here's a more recent example. There's a white tiger. Technically, this is not an albino. This particular mutation is called leucistic. The very last wild-caught white tiger was captured in 1951 in India and was named Mohan. There's the pedigree. I don't know if you can see it all that well, but up at the top, Mohan was bred with an orange female tiger whose name was Begum, and they had a bunch of cubs. The cubs were presumably heterozygous, and orange is evidently dominant to white because the cubs all came out orange. But Mohan was bred to one of his daughters. His daughter is kind of down towards the center. Her name was Radha. And when Mohan was bred to his own daughter, Radha, the offspring were homozygous and white. And then Mohan was also bred to his granddaughter, Sukeshi, which is getting down there towards the lower left. And that lineage has been maintained ever since by zoos and by animal breeders. And I don't know if you folks remember Siegfried and Roy. There were a couple of Las Vegas magicians that used a lot of animals in their acts, and they used to have white tigers. So we now have this breed of white tigers that's been maintained, kept in zoos, circuses, and the kinds of people who think it's really cool to have their own tigers. And this is why the Association of Zoos and Aquaria actually banned its members from breeding any more white tigers in 2011. Mohan's descendants are inbred, and they are homozygous for white fur, but a number of them are homozygous for very bad alleles, as well as the desirable white fur allele. So there's a high incidence there of tigers with birth defects. This is one of them. You can tell this tiger is, has got a definite problem. You can see the face is not developed the way that it should. So yeah, this is why, I guess they will probably persist in, dodgy zoos and roadside attractions and the kind of people who think that owning white tigers is an awesome hobby. Um, but yeah, reputable zoos and aquaria no longer breed white tigers for exactly this reason. It's all the fault of that bitch Carol Baskin. Sorry, sweetie. Real quick, this just has some pictures. I was talking about this last go around. Inbred mice have been mated brother to sister for at least 20 generations. They're homozygous at virtually all of their loci. They're virtually clones of each other. And researchers can experiment on inbred mice without genetic variation getting in the way because they're all genetically nearly identical. Some of these strains have been mated brother to sister for hundreds of generations. They're all homozygous at every flipping locus. Many strains are homozygous for genes that mimic human disease. This particular mouse of strain NOD-SHILTJ, I don't know how you'd even pronounce that, but mice of that strain are virtually guaranteed that if you feed them a certain diet, they will develop insulin-dependent diabetes. So if you're a diabetes researcher, you might well study the disease or test treatments on mice like this because they're genetically identical and they're all guaranteed to develop the disease at the same time in the same way. Okay, so yeah, we talked about that a bit on Tuesday, but I thought I would just show you some pictures of mice. Here's the thing. A guy named George Shull discovered this in 1909. Corn is normally pollinated by wind, which means that it's hard to control what corn plants mate with which. So to do it, you have to go around in your cornfield and cut off the male flowers um, so that the female flowers can be pollinated only with the pollen that you want. And so you can create lineages of inbred corn. And he found that when two inbred lineages were crossed, the offspring often came out larger and healthier than either parent. Up there at the top, that's corn plants growing in a field, and then you see ears down at the bottom. MO17 is an inbred strain of corn. B73 is another inbred strain of corn. Cross them, and you can see that the hybrid offspring are taller, and the corn ears are much bigger than either plant. The word for this is heterosis. You'll also see it called hybrid vigor. This is true for many other crops now. I assume you've seen grape tomatoes before, these tomatoes that are about the size and shape of a grape. They were only introduced to the US market in 1997. They're not that old. And they are a hybrid known in the trade as Santa F1. There's one particular breeder, I think it was a Chinese company, that developed grape tomatoes, and they're the only ones that know what are the inbred strains that have to be crossbred to get a grape tomato plant. So if you want to grow grape tomatoes, you have to buy grape tomato seed from the company that knows how to produce it. So the identity of the parent strains that you'd have to cross to get a grape tomato plant, that's a carefully guarded corporate secret. Time was that farmers used to save a portion of each year's grain crop as seed grain for the next year's crop. There are still farmers and gardeners that do this. If any of you are interested in starting a garden this year, there is actually a group called Seed Savers that maintain a lot of strains of vegetables, so-called heirloom tomatoes and things like that. And they'll show you how to isolate your seed and save it to plant next year. That's the way people did it for thousands of years. And it's what was being done as far back as the 1910s. You can see on this poster that Uncle Sam is warning the farmer to make sure that that farmer saves some seed corn. But we don't do this now, at least in large-scale operations. Beginning in the 1930s, farmers were encouraged to purchase hybrid corn seed. Seed producers like DeKalb would maintain purebred lines of corn, namely inbred lines of corn, and then cross those together to get hybrid seed. And then the farmer buys the hybrid seed and gets hybrid plants. Hybrid corn is often not just more vigorous, but it's a way that you can blend desirable traits from two different strains. So if you have one inbred line of corn that's big and a second inbred line of corn that is resistant to disease, but maybe not so big, with any luck, you can get hybrids that will be both large and be disease resistant, or in this case, be the golden snitch. This picture here shows what you get when you cross corn with the golden snitch, I guess. I don't know. I mean, it's got wings and it's golden, so that clearly must be what it is. That's responsible for dramatically increasing crop yields. This graph, the y-axis is tons of corn produced per hectare. Hectare is 3-point-something acres, I think. And OPV stands for open pollinated varieties. That's the corn where you save the seed and plant it from one generation to the next. And you can see that as different types of hybrids have been introduced, all of those open circles and squares and diamonds, crop yields have gone from being about 5 tons per hectare to about 9, almost 10. We've almost doubled corn production between 1930 and 2005. The catch, though, is that farmers now depend on seed companies to produce their seed stock. You can't save your seed anymore. You have to buy it because seed from hybrids is not going to breed true. If you take a hybrid plant and you save seed from it, the offspring are going to have all kinds of allele combinations. And, it's pretty much unpredictable what sort of offspring you might get. It's not an efficient way to work. So the price is that farmers are now dependent on seed companies to produce the seeds that they plant from. So there. Talk a little bit more about agriculture. I said way back that populations had to be large to be in Hardy-Weinberg equilibrium, and I was kind of vague about what exactly I meant by large. Now we can get at this a little bit more rigorously. Random effects in a population cause allele frequencies to fluctuate, a phenomenon called genetic drift. The example I always think of is, okay, eye color is more or less genetically determined. And we could, if we wanted to, go out to Little Rock and calculate the number of people with brown eyes and the number of people with blue eyes. Okay, and if we wanted to, we could try to calculate allele frequencies in the population of Little Rock. Now, people are always being born in Little Rock and they're dying in Little Rock. But in a large population, each individual birth or death doesn't change the allele frequencies very much. If one blue-eyed person happens to get run over by the River Market streetcar, That's a tragedy, but Little Rock's population is what, 200,000 or something like that. You know, one person happening to get killed is not going to have a big effect on allele frequencies. One blue-eyed person having 12 kids because they married a Duggar is not going to have a big effect on allele frequencies. On the other hand, if you drive up Highway 7 north of Russellville, you come to a spot called Booger Hollow, whose population, there's a sign out there that I think says population 7, counting one coon dog. So let's say there's 6 people living in Booger Hollow. Let's say some of them have blue eyes and some of them have brown eyes. And if one blue-eyed person in Booger Hollow gets run over by a dually rolling coal or whatever you drive up on Highway 7, that's going to have a much larger proportional effect. So in a big population, there's always random events that happen. You know, there's always members of a population that, for no particular reason other than dumb luck, happen to have extra kids or maybe die before they have offspring, for reasons that have nothing whatsoever to do with selection. In a big population, all those random events have very little effect, and many of them will cancel each other out. In a small population, these have a much larger effect. You should recognize these by now. Those are some curves showing allele frequencies over time in, I think, the same simulation that I had you folks work with. And you should have seen this by now. I set this to not have any selection. All of those W's were the same. And we started with one allele with the allele frequencies of 0.5. When the population was large, you can see that even after 50 generations, those lines showing the population allele frequency trajectories were still staying fairly close to 0.5. When the population was 20, you can see there's a lot more variation, a lot more fluctuation there. All right, I am assuming that all of you have observed something like this for yourselves. Okay, how much effect genetic drift has depends on the size of the population, but we can't just count noses. We can't just use the actual population size, because at any given moment, a real population may have many members that don't breed. They could be too old, they could be too young, they could be too sick, they could be unattractive. Well, I'll show you what I mean by that in just a second. We can come up with a concept called the effective population size, and that is the size of an ideal population that would have as much genetic drift as the real population that you're studying. An ideal population would be a population in which every member gets to reproduce. So what is the size of the ideal population that would have as much genetic drift as the actual population that you're studying? Now, there's different ways of calculating this. Don't memorize that formula that I've given you. If you have a population that includes breeding males and females, you calculate the effective population size as 4 times the product of the number of breeding males and the number of breeding females, all divided by the sum of the number of breeding males and breeding females. But I'm not going to ask you to do that. Let me show you what I mean, because the effective population size is not always at all close to the real population size. Here's a couple of northern elephant seals. These are a couple of males. They've got these big sacs on their nose. You may be able to see that they're quite scarred on the neck because they're fighting. And they're fighting by rearing up and exposing their big canine teeth and then bashing into each other with their teeth. This is the way that they fight. The fighting is sometimes lethal. And northern elephant seals have rather interesting social structure in that a very few males mate with almost all of the females. You might have just a couple of males, each of which might have a quote harem of 10, 20, 30 females that he and he alone mates with. So you might have, in a colony of northern elephant seals, you might have 2 or 3 dominant males that are doing almost all of the breeding, and the great majority of the males are not doing any breeding at all, which kind of sounds like my high school, but let's not get into that. The males that don't have harems challenge the males that do. That's why the males are constantly fighting like that. And if a male wins, the winner takes all. That male immediately gets to start mating with all of those females. But at any given time, only a very few males are mating with the females, and that means the effective population size may be surprisingly small. This is another case where you have a very low effective population size. Time was, if you had a herd of cows, you had mostly female cows because those give milk and they don't try to kill you. But you'd have a bull in the pasture just to keep the cows happy, as they used to say. The thing is that now that we have artificial insemination, it's possible for a cattle breeder to order frozen sperm from a champion bull. And that means that a single bull may father tens of thousands of offspring as his semen gets collected, frozen, and shipped all over the country. This is actually fairly big. This is a fairly big business. You can order champion bull semen if you are so minded and use it to inseminate your cows. And the upshot is that many cow breeds have an extremely small effective population size. In North America, Holsteins, that's a Holstein, they're the top dairy breed. Most of your milk comes from Holsteins, and they might have a population of several 100,000. I'm not even sure how many Holsteins we have, but it's quite large. The problem is that they're all being inseminated and bred using frozen semen from a very small number of elite bulls, and the effective population size has been calculated at about 80. So the actual population of Holsteins is showing as much genetic drift as a population of cows where all of the males and females were breeding that was 80 individuals would have. I think I managed to make that sentence come out making sense. The reason this is potentially a point of concern, go back here, if you have a small population size, you have a lot of fluctuation in allele frequencies. If you look at the graph down below, everything is just going zigzag, zigzag. If you look carefully, you'll notice that the red and the green lines zig and zag and zig and zag, and by about generation 25, they've hit the 0 line. What that means is one of the alleles has been completely lost from that population. The more genetic drift you have going on, the greater the odds that an allele is going to disappear purely by dumb luck. This is, in statistics, we call this a drunkard's walk model. If you imagine a drunk guy who's staggering down the street, there's gutters on either side. He might zigzag to the left and zigzag to the right, but eventually, given enough time, he's going to fall into one of those gutters. The same thing is going on here. Those allele frequencies may zigzag to the left and they may zigzag to the right, or zigzag to the top and zigzag to the bottom. But as soon as an allele frequency becomes 0, it means that allele is gone. And as soon as an allele frequency becomes 1, it means that allele is the only one present. We say that that allele is fixed. And so if you have a lot of genetic drift in a population, what you're risking is that population is going to start losing alleles from its gene pool and become less genetically diverse. And that often means that the population is less resilient, has less genetic diversity to draw on, especially if environmental conditions change. That population is going to be less diverse, less well able to respond to future selection. Okay. Oh, wow. I'm almost out of time, and those are a whole bunch of formulas that I'm not going to require you to memorize at all, except you can't directly calculate the effect of genetic drift because it depends on random effects. But the variance in allele effects is equal to P times Q divided by 2 times the effective population size. So the smaller the effective population size, the greater the variance is going to be. The more those allele frequencies are going to tend to spread out away from where Hardy-Weinberg equilibrium predicts they should be. Okay, that was kind of a lot. Please do not try to memorize any of those equations except for that way that we have of calculating the inbreeding coefficient is 1 minus observed heterozygosity over expected heterozygosity. And I will stop the recording now.